All right, so we're talking about photosynthesis. There are two stages of photosynthesis. One is light dependent and one is light independent. We will refer to these as photosystem 2 and photosystem 1. First, we're going to look at a cross-section of the leaf and point out a couple of important parts. The cuticle is just a waxy coating to protect the leaf from dehydration. These are found to be very thick in the evergreen trees, such as pine trees and magnolia trees, and are also found in desert plants to hold in water. The epiderma, there is the upper and lower epidermis. These are just for protection, sort of like your skin protects the rest of your body. The palisade mesophyll. The palisade mesophyll is a very critical part for photosynthesis. The cells are very, very dense with chloroplast, and this is where a lot of the photosynthesis occurs in the leaf. The spongy mesophyll also has some photosynthesis occurring in it, but it mostly allows for gas to move down, around, and out of the leaf. The stomata here are kind of like doors. They allow gas to flow through the leaf without losing water or carbon dioxide or any of the other molecules that the plant is going to need. All right, so photosystem 2 is the light-dependent reactions. It's called photosystem 2 because we discovered it after we discovered photosystem 1, or the light-independent reactions. But it does have to happen first. These reactions are called light-dependent because they do require the sun's energy in order to function. They happen inside of the chloroplast of the plant leaf, specifically in these pancake-looking discs here called thylakoids. There are three products produced by the light-independent reactions. These are NADPH, oxygen gas, and ATP. NADPH and ATP are used in the Calvin cycle, or the light-independent reactions. Another important part of the chloroplast we're going to look at is this part here called the stroma. The stroma is all of the space inside of the chloroplast, but outside of the thylakoid membrane system. We're going to talk about it more when we get to light independent. So the first thing that has to happen in photosystem 2, or the light-dependent reactions, is that the sun's energy is absorbed by the chlorophyll of the thylakoid. So chlorophyll is found on the inside of the thylakoid, in here. This here is showing you the thylakoid membrane. The energy energizes or excites the electrons. You can see in this situation you have that 2e minus. That is telling you that you're looking at 2 electrons. The electrons are going to enter into a chain of proteins called an electron transport chain. And as it passes through this chain, energy is being released from that excited electron. This allows two things to happen: photolysis, or the use of energy from the sun to split water, and hydrolysis of a phosphate onto ADP to form ATP. So the sun energy energizes electrons that are found in the chlorophyll here on the inside of the thylakoid. Those electrons are then pushed into the electron transport chain on the membrane of the thylakoid here. And as it travels through the chain, it releases energy. That energy is used to split water apart and to add a phosphate onto ADP to form ATP. So, we just talked about how the phosphate ion, which looks like PO4 3-, is added onto ADP to generate that ATP. We are going to use this ATP to provide energy for photosystem 1 or the Calvin cycle. The second thing that's going to happen is that water is split to form hydrogen ions and oxygen gas. In this situation, oxygen gas is a waste product that will exit the leaf. The hydrogen ions are going to be bonded to the NADP plus in order to form NADPH. These are called coenzymes. They are going to be used in the Calvin cycle as well, and you're going to see these again when we talk about cellular respiration. Here is the Calvin cycle. It is also known as photosystem 1 because it was found before photosystem 2. However, it has to have the products of photosystem 2 in order for it to occur. So you have 3 steps that are happening here. You have carboxylation, you have reduction, and you have regeneration. Carboxylation is when you get carbon dioxide reacting with ribulose biphosphate, or RUBP, to form an unstable 6-carbon molecule. This 6-carbon molecule will not stay stuck together because it is unstable. Therefore, it will split into 2 3-carbon molecules that we call PGA. PGA is going to go through reduction. This reduction uses the energy found inside of ATP and the coenzyme NADPH in order to form pGal. At pGal, our cycle splits a little bit. 1 out of every 6 pGal molecules is going to move into the cytoplasm here in order to form carbs. This is where glucose is being formed, out in cytoplasm. The other 5 pGal molecules are going to go through what's called regeneration, where we use the energy stored inside of the ATP in order to form or reform that RUBP we already used. This is known as regeneration because we are trying to get that RUBP back that we've already used. The fact that the pGal regenerates RUBP and that RUBP is needed for carboxylation to occur shows you that this is a cycle. It's moving around constantly, and it has to have each part in order to continue moving. All right, so review. Photosystem 2 are the light-dependent reactions. They are occurring within the lumen and specifically the membrane of your thylakoid, right through here. Photosystem 1, also known as Calvin cycle or the light-independent reactions, occur outside of the thylakoid in the stroma. So if we are looking inside of our chloroplast, here is photosystem 2, and out here in the stroma is photosystem 1. So this is our stroma happening in photosystem 1. This sugar is going to move out of our chloroplast and into our cytoplasm in order to form glucose and any other carbs that we need. And all of this is primarily occurring in this layer here of palisade mesophyll. So if we look at what we see, this is a leaf that we can see. When we put this leaf under a microscope, we can see inside of it and we'll see things like this. Then, if we use a very powerful microscope to look at one of these palisade mesophyll, we will see a chloroplast that looks something like this. Then, if we decide to focus on a stack or granum of our thylakoid, we will be looking at this. This here is a granum, which is just a stack of thylakoids. If we look at just one thylakoid, we will see photosystem 2 occurring. If we zoom out and look in the stroma, this is where photosystem 1 will occur in the stroma. And our sugars are going to occur outside of the chloroplast in the cytoplasm of the cell. So that's outside of this in the cytoplasm of one of the palisade mesophyll cells.